Okay, welcome. This is a review of Chapter 4, Tissue Level of Organization. The study of tissues is called histology. And for our investigation of this, we will move forward in anatomy and physiology to review the different types of tissue. So let's get started. Now, we've already talked previously about the different organization levels or the hierarchy of organization. Chemical level, you basically start with atoms. Then you go to molecules. Then you go into structures that make up the components of a cell. Then you go into cells. Excuse me. Now we're going to take and look at tissues. A tissue would be composed of a cell of a particular type and many of these cells. Now, it is tissues that are really the building blocks for organs. There are four major types. Epithelial, which covers exposed surfaces, whether that's internally or externally. It can form secretory glands and lines internal passageways and chambers. Connective tissue, which fills internal spaces and provides structural support. It also stores energy. Hint, adipose tissue. Nervous tissue contracts. Oops, they had a typo there. It's supposed to be it's supposed to be muscular tissue there. And muscular tissue contracts to produce movement. This includes skeletal muscle, cardiac muscle, smooth muscle. Now we get the right one here. Nervous tissue conducts electrical impulses, carries information. It is one of the means in which you are able to communicate throughout the entire area of the body. And we'll get more into this as we move forward. Now, the thing is, we need to see tissues, and in some cases that gets into the microscopic level of structures. So we have to use microscopes. When we look at the person, the unaided eye, you can only go so far, about maybe down to about 1 millimeter. But with a compound-like microscope, you can go in the range from 10 millimeters down to about 1 micrometer, somewhere around there. Now, a micrometer, to help you, micro refers to 1/1,000,000th of a meter. A millimeter, the milli prefix, refers to 1/1000th of a meter. But there are structures that are even more sophisticated and finer in detail, and we can't see them with a compound light microscope. That's why we get into use of electron microscopes. The two major types, scanning electron microscope, and that can go down to about in the range of 100 nanometers. And for those of you that are wondering, what's a nanometer? Nano refers to billionths of a meter. That's where it is possible with scanning electron microscopes and other types of electron microscopes, such as the transmission electron microscope, that you can see structures like viruses and even subcellular structures like ribosomes and even some proteins. Transmission electron microscopes can go right down to the level of almost amino acids, and that's at 1 nanometer. In other words, a billionth of 1 meter. Now, what I have done for you, and I encourage you to print these out, they are available in your Blackboard, are concept maps. Concept maps are basically set up in various ways to provide a visual understanding of certain amount of information. They make it easier to kind of help memorize, especially if you get a lot of different details, to kind of put things into an order. In the way I did these is a comparison and a contrast, and it becomes almost a logical question. So if we look at the differences between epithelial, connective, nervous, and muscle tissue, we ask a couple of questions first. Does the tissue have a free edge, meaning is it exposed to a lumen, either internally or externally? If it's yes, then it's epithelial. If it's no, then the next question is, does the tissue cells have contractile elements? If the answer is yes, then you have muscle tissue. If it's no, then you come down to a final question. Does the tissue have a large amount of extracellular matrix? If it's yes, it's connective tissue. If it's no, then it's nervous tissue. For the types of microscopes, you need to be aware of a couple of differences and limitations. Compound light microscope, fantastic, and relatively affordable in a basic laboratory situation. It uses light waves, can get up to a magnification of about 2000x. Now that means magnification of 2000. The electron microscopes differ in that they use the wavelengths of electrons, which is much more finer in detail. You can get up to about 500,000x. But there are some very great limitations. And by the way, when I say transmission versus scanning, think of it as like looking on the exterior of the structure. Transmission can go right into the structure. Each of them requires that the sample be dead. Now, why? First, you have to view it under a vacuum. Any gas particles would cause the electrons to be deviated from actually scanning your target. Second, a lot of the samples have to be frozen to be properly scanned. Third, some of the targets have to be coated with a thin layer of either tungsten or gold to basically be capable of having the structures viewed, and the electron beams would bounce off of these substances that coat the structure. So in a nutshell, what do you get? Compound light microscope gets lower magnification, but you can view items in color, alive, and you can actually make videos of it. Transmission electron microscopes and scanning electron microscopes have a much greater magnification, but you cannot view anything in video, in motion. It is purely black and white or basic grayscale. If you see any colors in an electron microscope, that's been computer enhanced. That's all done after the image. Okay. Also, the target, whether it's tissue or a microorganism or anything like that, they're all dead. Okay. Let's take a look at these. This is all epithelium from the respiratory tract. Now, if you notice here, this is using light microscopy. And the thing is, we had to stain it with specific chemicals so that structures such as the nucleus and the cell membranes would be able to be visible. Otherwise, it's like looking through different layers of glass. Now, these are all epithelium of the respiratory tract, basically the same structure. So LM is light magnification, light microscopy, and it's at a magnification of 400. The epithelium, yes, this is again same respiratory tract, but it's using transmission electron microscopy at a magnification of 3000. So that's even greater than the maximum you could get with light microscopes. You'll notice the various structures that are here, okay? And you get a much clearer image, although it is black and white and the tissue is, of course, dead now. Now, here's something that's even more fascinating. What they did was they took the epithelium. Remember the cilia that were up on the top here on the epithelium? Well, they were able to take it at a different view using a scanning electron microscope. So the electron beams were aimed in a way to bounce off the exterior of the structure to give you views such as the cilia that you see there. This was at a magnification of 15,846, but, and this is the important point, that's not the natural color. That was all color enhanced after the image was taken using a computer program, okay? So, now that we've gone through all of that, let's start moving into the different types of tissues. Epithelial tissue will include epithelial as well as glands. The glands that we're going to be talking about include both exocrine glands and endocrine glands. Exocrine have ducts, passageways, or ports out into the lumen. Endocrine glands do not. But let's move forward. What are the functions of epithelial tissue? To provide physical protection, control permeability, provide sensation, and produce specialized secretions. Okay? Now, what is very common in different tissue types are structural elements. Here, epithelial tissue has unique structural elements. They will either be extensions on the cell membrane, and there's a difference between one side of the cell and the other. But we'll go through those parts in a minute. You will either have extensions of the membrane called microvilli. This increases the absorptive surface area of that surface, or you may have actually extensions that propel substances past it, such as motile cilia. They will beat and move liquids or materials by, such as what you see in the respiratory tract in the trachea. What you're going to notice is that the two differences on the surfaces here. So apical is one side. That's the side that usually faces the lumen or the space out exterior. And then there's what they call a basolateral surface, which has a basement underneath. This is to prevent the structure from being sloughed off or shed. Now you'll notice that you have a nucleus, endoplasmic reticulum. You'll have a number of mitochondria, which of course are producing ATP. You'll have a Golgi apparatus here, which packages the products, and then they are sent up to here, the apical surface, for release. Now, there's different types of epithelial tissue. There is simple epithelia. To help you to discern between simple epithelia and stratified, think of the term simple, basically one cell layer thick. Stratified comes from strata, meaning layers. So with squamous, you'll notice that there's sort of a central nucleus, but it's sort of flattened. And so there's very little cytoplasm here, okay? It's almost like tiles that you'd see on a floor. Cuboidal, the cells are cube-like, and they have a central nucleus. They have a larger amount of cytoplasm in the volume here. This, of course, is basolateral. This, of course, would be the apical surface. And then columnar. Now, columnar, think of the word column. So they're sort of rectangular. They have a central nucleus. It's either going to be somewhat around here, slightly toward the bottom, but not a lot, or centralized. But one thing that's unusual is that the nucleus is not round or spherical. It is more oval in shape. Now, it's stratified. What you always have is at the basolateral side, these cells right here are the ones that are going to be undergoing mitosis to divide. And as they do, the cells are pushed upward. And as they are pushed upward, they go through several morphological changes, morph meaning shape. And as they do get moved up closer and closer, the reasoning behind this stratification is a lot of times the cells right here are sloughed off or knocked off after a time. So this is the apical end, this is the basolateral. Cuboidal has its own stratification, just not as many cells. But you can see they're somewhat cuboid or cubic, square-like, okay? Columnar is interesting because here's the basolateral side, here's the apical side, and what you notice is in the beginning, it looks almost as if it's starting cuboidal, but then it goes through these morphological changes and becomes more columnar. The nucleus, instead of being round and at the center, becomes more oval-like, and it may either be in the middle of the cell or slightly lower, okay? Epithelial cells are interconnected structurally and functionally. In other words, so when you have these cells connected, they have to form sort of a solid, protective layer. And so what provides them these solid protective layer structures are what you see right here. This is the apical surface. This is the basal or base surface. You have unique structures that attach cell to cell. Tight junctions are one. These are interlocking membrane proteins that basically are like rivets, okay? And so they basically attach one cell membrane to the neighboring cell's membrane. Another type is called an adhesion belt, where cytoplasmic fibers are inside the cell, but then they attach to these unique structures here and here. And these structures provide sort of a limiting barrier by which microorganisms or any other substance could pass between the cells. Finally, you start having these other unique structures, desmosomes, which you can see there's an internal anchoring of cytoskeletal elements. Then there are some unique proteins here, cell adhesion molecules, CAMs, and it's almost like Velcro. The CAMs attach to one another. They're filled with some hyaluronic acid in between, and basically they have this plaque-like protein out here and here that basically gives them an anchor, and so they're really tightly connected together. Now, we have desmosomes, and then we have this other unique structure called hemidesmosome. You can see somewhat the similarity, but the difference. This is providing using intermediate filaments and then your plaque structure, and then what happens is that you have some unique proteins that reach down here and basically attach to not merely the basal lamina, but the reticular lamina. In other words, it allows them for a solid anchoring to the basement membrane that is beneath them. That's why they're called hemidesmosomes, because they don't have two desmosomes, only one. And that's sort of an anchoring structure. Okay? So, tight junctions, adhesion belt, and finally, also a means to have cell-to-cell communication. You're going to find this very important down the road, especially when you talk about cell-to-cell communication with cardiac muscle cells. Connexins, and that's a protein, basically form these tunnel-like structures between one cell and the other. In doing so, it allows for the flow of ions or other substances, particularly for synchronization of certain functions along all the cells at the same time, such as the contraction of muscles. So you do need to review all of these different types of interconnections that you have. And just to expand upon this, tight junctions, they prevent water and solutes from passing between the cells. You can see where the interlocking membranes are, the proteins. The adhesion belt is a continuous belt around the cell and binds the neighboring cells. This consists of dense proteins attached to the microfilaments inside the cell, as you can see here. Connexins, this is the interlocking transmembrane proteins. They allow the passage of ions to pass for cellular synchronization. Then we get CAMs, the cell adhesion molecules, that play a very important role in desmosomes, okay? These bind to each other and to extracellular proteins, as you can see right through here. The hyaluronic acid, this is a polysaccharide derivative that works with proteoglycans to assist in membrane binding. So you see this all the way filled with hyaluronic acid. Now, as I mentioned to you before, the hemidesmosome attaches to the deepest layer of the epithelial cells, basically to the basement membrane, as you can see down in here. So whenever you have what's called a basement membrane, you're going to have a basal lamina, which is right up close and next to the epithelial cells. And then a lot of times a reticular lamina occurs below that. So it's really anchoring itself down to the lower levels of the basement membrane. Now, where are you going to see some of these cells? Well, let's start off. Squamous epithelial cells are thin, flat, irregularly shaped, and they make sort of a nice thin layer or coating. You will see them in the peritoneum, as you can see over here. Underneath them would be connective tissue. And you have these sectioned epithelial cells that you see here. Notice that they have their cell membranes connected. as sort of puzzle pieces right next to each other. A central nucleus, the cytoplasm is very thin in volume, and basically the nuclei is present here. If you see any shadowing here, this is nuclei basically supporting the underlying connective tissue here, okay? But squamous epithelial cells, this is going to be now, when we talk about the stratified, this is where the uppermost layers are shed off, and you want to know where some of the locations are, for example, in the cheek, on the surface of the tongue, etc. Here's an example of this, the stratified squamous epithelium, light microscopy 4000. You see here is all the stem cells. Now, stem cells are basically the cells that go through mitosis to replace cells above them, okay? So you have a basement membrane here, you have the connective tissue here. These cells will progress, change, have their biochemistry altered a little bit until they become much more flattened out and eventually are easily sloughed off or shed off. If instead these cells fill up with a particular type of protein called keratin, we call them keratinized. The apical layers are filled with the filaments of keratin, which is a very water repellent but very resilient protein. This is where you see them. Keratinized skin cells here in the upper levels of the epidermis. The keratin fibers kind of form sort of these coiled cables, and usually as you're going through the development, the stem cells are down here, and they will go through several levels of progression until they basically die out, but they're filled with keratin, okay? If we say that they're non-keratinized, the cells are present to basically resist abrasion, but they can dry out, okay? Cuboidal and transitional epithelial cells are very interesting. The cuboidal are, when they're simple, they just resemble sort of hexagonal boxes. The nucleus is in the center. You have limited distribution, but this provides for secretion and or absorption at that layer. When you get stratified cuboidal, this is rare also, but it exists in the sweat gland ducts and in the mammillary gland ducts. For transitional epithelial cells, they're very unusual. Where you're going to see them best known is in the urinary bladder. Because what happens is that in the urinary bladder, you have these repeated episodes of stretching and recoil. The bladder fills up with urine and then contracts down as we expel the urine. But when you look at the relaxed state, all the layers, which can be anywhere from 3 or 4 to about 6 cell layers in thickness, are somewhat kind of rounded, etc. But when they all start stretching out, just like filling up a balloon with air, you're going to see that the cells, particularly the top ones, are going to kind of thin out. It's almost as if they're being stretched out, but they will recoil back when you empty the bladder. You can see here simple cuboidal epithelial cells. These are well known for secretion absorption. They're in the nephron here, which is a structure that's key in the kidney. So you have on each side of these sort of connective tissue, you have a basement membrane, and you have simple cuboidal cells here. This is the empty space, the lumen of the duct, and of course you can see the nuclei of each of the individual cells here. When we look for stratified cuboidal epithelial cells, here's where you got it. And of course, you're going to have the lumen of the duct right here and right here, here. You're going to have the several layers, 1, 2, 1, 2, 1, 2, etc., of the cuboidal cells. And you will have a basement membrane on the kind of outer exterior of each of these ducts, DUCT, mind you, okay? And you have some connective tissue in between these ductworks, but basically what you've got are just multiple layers here that are involved in processing the liquids and conveying this material in the sweat gland duct. Here's what I was talking about earlier. If you have an empty bladder, you'll notice that the epithelium, which is not stretched, you've got somewhat kind of rounded cells, and it may be 5, 6 layers thick. Notice at the top even is kind of round and bulbous, but underneath it, you have the connective tissue. You also will have underneath that a smooth muscle layer. As you start stretching out, as the bladder is getting filled up, what happens? The epithelium will get stretched. You'll still have basement membrane, you still have connective tissue, still have smooth muscle layer, but you'll notice that the cells start taking on, instead of a kind of bulbous or rounded appearance, start flattening out and thinning out. And that's the transition, to make a change, to help you to understand that. Now, columnar epithelial cells that appear rectangular, they're found where absorption or secretion occurs. You'll see this with stomach, small intestine, gallbladder, nephrons, and the kidney ducts. The nucleus is usually near the basal side. Microvilli enhance the absorption. Cilia propel substances past the apical side. Now, there is another unique form called pseudostratified. Pseudo refers to false strata. They have an irregular array that makes it appear as if it's stratified, but it's not. It is simple, but it's such a jumbled mess that if you look at it from an angle, such as what we have with any slide, they appear falsely stratified. Here is an example of simple columnar epithelium. You would find this in the intestine. Microvilli here to enhance surface absorption. This would be the lumen up here. You've got a large amount of cytoplasm here. The nucleus, which is more oval shaped. Underneath here you have connective tissue and a basement membrane right there, okay? But when you go into the trachea, nasal cavities, larger lung airways, you have the appearance of pseudostratified. Now, if you look at this just from the angle, excuse me, this appears as if there's just cells on top of cells on top of cells, but it's not. If you follow any of these extensions, it's basically they're twisted and jumbled around in a certain way. But basically, if you take a look at this cell from the top all the way down, it has a bottom attached right at the basement membrane level. And you can see this. You will also see the cilia at the top. These would be moving, they would be beating along, moving liquids, particularly mucus, through the area up the trachea. Okay. Now, stratified columnar epithelial cells, rare. You see this in the salivary gland duct. Here's the salivary gland duct. It's like they took a cut right through it. This is the lumen. This is where the saliva would pass through. Notice that you have the deeper cells. They're going through the mitosis to make new replacement cells here. But finally, you have the columnar structure, the change in morphology, the change in biochemistry that faces the lumen here. Okay, so cytoplasm, nucleus. You've got the lower levels here. You have a basement membrane here, and you have this being stratified. Now, glandular epithelia is another area that's really important. When we talk about collections of epithelial cells or structures derived from epithelial cells that produce secretions, we end up with two major types, exocrine, which is with a duct, or endocrine. There's no duct at all. Usually what happens there with endocrine tissue is they deliver their product directly to capillaries, which are then distributed by the blood to a variety of other organs and tissues, etc. Exocrine will make a particular material, but then it will pass through a small ductwork and eventually out to somewhere else. For example, the pancreas. The pancreas is also interesting in the sense that it's both exocrine and endocrine. It has little patches of cells called the islets of Langerhans that will produce insulin, glucagon, somatostatin, etc. And that is the endocrine part, because when they secrete these hormones, it is transferred right into the capillary bed to be distributed throughout the body. The exocrine part, which consists of digestive enzymes and bicarbonate ions, will be basically put into a ductwork that eventually empties this solution into the duodenum of the small intestine. Now we need to talk about also types of secretion. This is a method of delivery. Now, you might think to yourself, oh, it's just all exocytosis. Oh, no. Different secretions occur using different mechanisms. Merocrine is basically your secretion via exocytosis. So you have these substances in vesicles. The vesicles will fuse with the apical side of the cell membrane and release their contents outwards. Apocrine is a loss of the cell cytoplasm. So huge or large sections of the cytoplasm are released, which contain vesicles of the secreted product that's to be occurred. Holocrine is even more interesting because that gland cell will destruct, and as it does, it will release all these vesicles containing a certain product. So you've got three ways. Merocrine, you can see these little vesicles leaving the Golgi apparatus, and in this case it would be mucin, which is a protein, which when you mix it with water, it's mucus. And you have the secretory vesicle fusing here. This would be exocytosis. Okay. Apocrine, the lower section or the upper section of this particular cell basically pinches off, releases the cytoplasm blob with the vesicles, and it is released out here. The cell will regenerate, regrow this membrane level and cytoplasm and continue to produce more of these vesicles that will eventually be pinched off and released. Now that's apocrine secretion. What about holocrine? Where you see holocrine, interestingly enough, is in the production of sebum, in other words, the oil, your sebaceous glands, okay? So we've talked about merocrine secretion, you see it with the salivary gland. Apocrine, you see it with the mammary gland. But in holocrine, the cells fill up with a certain amount of these, the cytoplasmic contents, and the whole cell just comes apart. The cell bursts, releasing its cytoplasmic contents, okay? The cells beneath it are being replaced, as you can see here, by mitosis. And as the cells change, it's a perfect example of changing in morphology, changing in structure and function. They fill up with what they're going to secrete. In this case, it would be sebum, oil. And as they get to the basic lumen area, they burst. One other thing about unicellular glands that act in sort of a releasing process, goblet cells, you'll see these particularly in areas like some areas of the gastrointestinal tract, some areas of the trachea. They're single cells that are mucus cells. They secrete mucin. Now, after you leave that, for unicellular glands, you've got other cells that are more multicellular in their approach as exocrine glands. And the classification of these is dependent on the structure of the ductwork. Is it simple? Is it compound? As well as the shape of the secretory area of the gland. Is it more tubular or is it more sac-like? And this is when we refer to the sac-like pockets as acinar. Now, the relationship between the duct, simple or compound, and secretory area, branched or not, gives us a grouping of identification here. When we talk about branch, by the way, this refers to the glandular area only. And it can get a little complex, I agree, but it's very important. A simple tubular, such as what you see with intestinal glands, here is the gland cell. Okay. No ductwork. Simple coil tubular, merocrine sweat gland. Here's some of the ductwork, and the rest of it is a simple coiled tubular structure. Simple branch tubular, Branched, 1, 2, 3. So there's branches here of the glandular cells. They still have a bit of a ductwork here. You'll see these with gastric glands, mucus glands of the esophagus, tongue, and duodenum. Now we start getting into acinar. It's more like a sac or a flask, okay? So you're going to have a longer, usually ductwork, and it comes as sort of like a cul-de-sac. You see this particularly in the stage of embryonic development of simple branched glands. You don't usually find this in adults, but the simple alveolar, the acinar, are present. Now, simple branched alveolar, where you have 1, 2, 3, and you still have the duct structures here. You'll see this with sebaceous oil glands. Now, what about if we had combinations of all these? By the way, this is our goblet cell. Notice how you build up right near the apical side a large amount of globules, vesicles containing mucin, okay? The compound tubular, you can see this, 1, 2, 3. So you see this in a variety of structures. The seminiferous tubules of the testes, the bulbal urethral glands of the male reproductive system, as well as the mucus glands in the mouth. The compound alveolar, the acinar, you see these with the mammary glands. So here's a flask here, okay? And in between here, you've got multiple ductworks here. And then you have what we call the grandfather of it all, the large combination of everything, a compound tubular alveolar. Here are some of the alveoli, the tubular structures. Here are some of the alveolar structures. Notice you've got 1, 2, basically 3 ductworks. It's a mixture. You see this with salivary glands, glands of the respiratory passageways, and the pancreas. Now, this is just sort of a concept map to help you because it can get it confusing, and I understand that. Here's where you start off with epithelial tissues with your concept map. First question, is the tissue a single cell layer? If it's yes, it's a simple tissue. So you ask yourself, are the cells flattened? Yes, simple squamous tissue. No, are the cells cuboidal? Yes, then it's simple cuboidal. If it's no, the next question defines the last couple of choices. Do all of the cells reach the free edge? No, pseudostratified. Yes, simple columnar tissue. With stratified, if you came up with no and you had stratified tissue, then you have these questions that help you to identify. Are the surface cells flattened? Yes, stratified squamous epithelium. No. Are the surface cells cuboidal? Stratified cuboidal epithelium. And if is the superficial layer only columnar shaped? If it's yes, then you have stratified columnar epithelium, but no, then it's transitional epithelium. So that's what you would see in the urinary bladder. Now, we're going to start moving into connective tissue. What is making them so different is they're surrounded by extracellular matrix. The matrix makes up the bulk of the volume of the connective tissue, so this is very different. There's a lot of secretion to make up this extracellular matrix. The connective tissue shares the following: specialized cells, extracellular protein fibers, and fluid ground substance. There are different types of connective tissue. To help you, loose, there's areolar, adipose, reticular. Dense, dense regular, dense irregular, or elastic. Fluid, blood or lymph. Supportive, either cartilage or bone. We're going to go through all of them, don't panic. Let's take a look at the connective tissue here. So you're going to have the connective tissue proper, and you have either loose or dense. So it's the arrangement here, and what is the amount of fibers versus the arrangement of cells. And by the way, remember in cells, you can have a whole host of them, but the key ones are going to be fibroblasts. Okay? Fluid. In this case, you have basically a fluid medium with certain selective cells inside that. With lymph, you're going to find a lot of white blood cells, etc. With blood, you're going to find an arrangement of erythrocytes, red blood cells, white blood cells, and we call them sort of cell fragments, are called thrombocytes, the platelets. Also, supporting connective tissue. So they've got to have something in an extracellular matrix that makes them unique. With cartilage, we're going to see this in a bit. 3 different types of cartilage: hyaline, elastic, and fibrocartilage, or bone. There you have a high deposition of calcium salts. Now, functions of connective tissue: to establish a structural framework for the body, to transport fluids and dissolved materials, to protect delicate organs, to support, surround, and interconnect other types of tissue. It's also available for a type function to store energy, especially in the form of triglycerides. Think adipose tissue, as well as defend the body from invading microorganisms, which we're going to get into in a bit. Here is sort of the all-purpose generic version of the loose connective tissue. And you got to keep in mind, loose connective supports other tissue types as well. Okay? What are you going to see in there for cells? Adipocytes, those are the ones that are lipid containing. It's almost like they're like fuel tanks. Notice that the nucleus is pushed to the side and it's filled with triglycerides here. A lot of times when you look at this under a microscope, because the staining procedure washes out or is lipid soluble, they tend to remove a lot of this lipid, so they look like empty tanks. They look like empty cellular structures. You have fibrocytes and fibroblasts that are involved with the construction of various fibers that make this up. The fibers that we have are going to be elastic fibers made of elastin and collagen fibers. There's going to be also a thinner version of some of these fibers that we call reticular fibers, because they're more, rete is the prefix that talks about a network or a meshwork. When we talk about fixed cells, these are cells that stay here permanently. So these would, of course, include adipocytes, fibroblasts, mast cells, fixed macrophages, melanocytes, which offer some pigmentation. Wandering cells are the ones that come in and leave, come in and leave. They may change into something and leave. Plasma cells, a form of white blood cell. Macrophages, another type of white blood cell. Mesenchymal cells are the early developmental cells. They may be there a little bit from an embryonic stage, but they mostly will eventually change into something else as time goes on. Neutrophils, eosinophils, another type of blood cell, as well as lymphocytes. Now, you see all this purplish stuff here? This is what we refer to as ground substance. We're going to get into some of this as we go into the loose connective tissues. The least specialized, but yet most common form found in adults is areolar tissue. So there's not a lot of specialization, but it's essential as a loose type of tissue to be useful in our structure. Adipose tissue, as I've said, supports and cushions. So therefore, you're going to find it in the buttocks, the orbit of the eyes, the abdominal pelvic cavity, and around the kidneys. See, here is the adipocytes as a whole cell. Yet what you find when you look at this under a microscope, notice all that empty space there. That's because all of the lipid, the triglycerides, were washed out during the staining period. And so what you see are basically cell membranes and the nucleus. That's it. Okay? Finally, reticular tissue, which you would find either in the liver, you may find it in the spleen, lymph nodes, bone marrow. It forms a tough, flexible network, and this network is usually called stroma. And what happens is that the fibers are there, and there's some cells that basically support the fibers, but they create sort of like, it's like building a house. Have you ever seen somebody who builds a house? You go inside, you know, the house on the outside looks nice. You got windows, you got doors, you got, you know, they may have even painted it, but what's inside? Nothing. There's no furniture. There's walls. There may be even electrical outlets and all that other neat stuff, but it's a shell. You got to put things in it, right? Well, any place that you see reticular tissue, you're going to have a variety of other cells present, residing in this reticular, almost like this spongy space. And they may be hepatocytes for the liver. They may be in the spleen, a variety of red blood cells and white blood cells, etc. We'll get more into that as we move forward in the course. Now, dense connective tissue is dominated by extracellular fibers. No way around it. The difference between dense connective, between regular and irregular, I should say, is the pattern of the collagen fibers. Remember I said the fibroblasts really play a key role in laying down the fibers? Here are your fibroblasts. Notice the waviness, yet the stackedness right next to each other, all along the same plane. This is from a tendon, okay? So this would be dense, regular connective tissue, orderly stacked pattern, located usually in cords, what we call tendons, or sheets, connect muscle to bone, or connect bone to bone when we talk about ligaments. Very, very strong, but usually in one direction or one axis only. Now, here's where we get into something more interesting. Irregular, dense, irregular. If you notice here, this is the dermis of your skin, okay, below the epidermis. And what you've got is patterns of collagen fibers, but they come into the plane of the sheet. They are basically following along. They're not stacked nicely. And they basically would go in the order of the x-axis, the y-axis, and the z-axis. So basically almost like coming out of the screen or coming out of the image. This irregular pattern, the interwoven meshwork, means that you can have a lot of stresses, whether it's push, pull in the different orientations, and it will recoil back and it will be fine. Therefore, this type of tissue forms coverings or capsule for visceral organs, superficial layer covering bones, cartilage and nerves, and a thick supportive layer of the skin that we call the dermis. Finally, we get into the elastic connective tissue. Now, why is it elastic? Really what it is that elastic fibers are now outnumbering the collagen fibers. So it has a lot of capability to taking stretch and properly recoiling back. And you can see this. There's certain elastic fibers here. You, of course, are going to have the fibroblast present. And see where you have this located? This is in basically an area of where you're going to have some ligaments. And the tissue is very, springy, resilient, and allows for extension and recoil. Okay, this is located between the vertebrae of the spinal column, the walls of large blood vessels, transitional epithelial and erectile tissue of the penis. So basically what I'm saying is material that can stretch outward and yet recoil and not suffer any damage, basically maintain shape and structure and function. Now, what about blood and lymph? Well, I'm going to tell you, if you go back about 20 years ago, they used to still consider that blood was considered almost like the fifth connective tissue. They called it sort of a fluid one, but now they just consider it part of the connective tissue. Lymph is the same way. The deal with blood is that it has what we call formed elements, suspended in a watery matrix that we call plasma, right? What are the formed elements? These are the red blood cells, white blood cells, platelets, okay? Blood is confined to the circulatory system, heart, arteries, capillaries, and veins. Now, lymph is formed as interstitial fluid that drains into the lymphatic vessels that drain into the venous system. But there's something that you need to keep in mind. Lymph is very similar because it also will contain some of the formed elements of various white blood cell types. Different types of lymphocytes. You'll find sometimes antigen-presenting cells. They are called dendritic cells. You will find NK cells, natural killer cells. You will find a variety of macrophages cruising along. And so it is important that you have recirculation to maintain homeostasis And so the fluid is taken from the extracellular compartment, put back into the blood, etc. But this is really critical also. Lymph provides the immune system with alerts to various types of infections. And also lymph helps maintain blood volume. Now, here's what you're going to find in blood. Platelets, they consider them almost like little bags of clotting medium. You got the white blood cells all the way through here, monocytes, lymphocytes, eosinophil, neutrophil, basophil, red blood cells. They're all important here. The lymphatic system, you're going to find not red blood cells, not platelets, but you will find a variety of some of these other cells, particularly lymphocytes, and also in some cases monocytes, macrophages, and other types of cells. As a result, they consider this a type of connective tissue. It's more of a fluid type of connective tissue. Now, let's talk about cartilage. This is a flexible support for the body. One key point, it is avascular, meaning it lacks a vascular system. It lacks capillary blood vessels in the cartilage itself. Okay? You're going to hear me saying this a million times. Get it down now. Cartilage is slower to heal than bone. That's because of the presence or absence of capillary beds. Okay? You break the tip of your nose, that's mostly cartilage. You break up near the actual frontal bone, you break the nasal bones here on the nose, they'll heal quicker than that cartilage damage. Chondrocytes make cartilage. Chondrocytes are located in lacunae. These are little spaces in the cartilage structure. The matrix contains polysaccharide derivatives, chondroitin sulfates, which form complexes with proteins to make proteoglycans. Now, by the way, you might have asked, avascular, so how does it live if it doesn't have a vascular network? All of the transport of oxygen, carbon dioxide, nutrients, waste, is by diffusion. So there are nearby capillary beds, but not in the cartilage itself. So cartilage is going to have a slower metabolic activity, hence a slower capability to heal, because it's really dependent upon diffusion of nutrients to allow for repairs. This is very different than how you have bone. We'll get more into bone in a few minutes. There are three major types of cartilage. Hyaline, which covers bone joint surfaces. Elastic, which contains elastin fibers. Thereby, it makes it very flexible. And fibrocartilage, this is where collagen fibers are present in the matrix, and therefore it resists compression, so stress on it. It's located in places like the pubic syphus, the knee joint, and the intervertebral discs. As you can see here, here's an example of hyaline cartilage. This is all this extracellular matrix produced by the chondrocytes, but they live in these little pocket holes called lignae. This is the hyaline cartilage from a shoulder joint. You can see this as the actual slide here. All of this is the matrix. Here are the lacunae, and in here are the chondrocytes. Now, the pinna, or external ear, is where you're going to see the best amount of elastic cartilage. Notice that in here, you still have the lacunae, you still have the chondrocytes, but what you have here is also quite a lot of elastic fibers, which allows for flexibility and recoil, okay? And you can see these stained fibers right throughout the entire network of the extracellular material here that we call the cartilage. Okay. Finally, as you can see here in this intervertebral disc, we have some rows, some structures of chondrocytes laid out in lacunae, but then you have layers upon layers of collagen fibers in the matrix, okay? And that can handle tremendous forces of compression or stress and strain. Now, cartilage, you have to understand, is covered by perichondrium. There are two layers, an outer fibrous layer, which is a dense, irregular connective layer containing fibroblasts, providing mechanical support, protects and attaches the cartilage to other tissues. An inner cellular layer that is important for growth and maintenance of the cartilage. The blood supply to the perichondrium supplies nutrients to the chondrocytes. Again, this is all based on diffusion now. The types of cartilage growth, there's two types, appositional, interstitial. Appositional is present in birth up to adulthood and can offer some repair. Okay, this is, as I mentioned, if an adult got their nose broken at the cartilage area, so you would have appositional growth. Interstitial is predominant in birth through adolescence. In adults, injury may trigger some repair. Remaining damage is a fibrous patch. Okay, so this is appositional growth. As you can see here, here's your fibrous layer. Here are the stem cells of chondrocytes that are undergoing mitosis. You've got the perichondrium here. You've got the chondrocytes there. Excuse me, these are the fibrous cells, so they're fibroblasts, not chondroblasts. They are undergoing, though, here the division. And what you will see is cells are going to differentiate into chondroblasts. So as time goes on, you see some of these cells converting from fibroblasts over into chondroblasts. The chondroblasts are going to secrete new matrix around this area that requires it. And once they get encapsulated here, they're going to be stuck as going from chondroblasts to chondrocytes. The direction of growth is basically, let's say, vertical, if you want to say it. It's in one dimension. Now, interstitial is a situation where the chondrocyte in the lacunae, surrounded by matrix, the chondrocyte undergoes division and it's in all different directions. Literally, the additional matrix secreted pushes the cells apart. So this interstitial, think about it, you talk about interstitial fluid between cells, fluid between cells. It's another way of saying extracellular fluid. So interstitial is literally between the cells, and when they start separating at the point of the lacunae, these cells will begin to lay down the new matrix. But you only see this from, let's say, adulthood to adolescence. Now, bony tissue, otherwise known as osseous tissue, is extremely important. 2/3 of the bone matrix is composed of calcium salts. Calcium salts in this case are calcium phosphate, calcium carbonate. The matrix is also composed of collagen fibers, which make it strong and flexible. See, here's the point. If you just had something that was made just of calcium salts and no collagen fibers, it becomes very brittle, it can break, etc. You have to have the proper laying down of the collagen fibers at all times. Now, bone is vascularized, meaning that it has blood vessels going throughout its entire structure. Therefore, it heals faster. Bone cell types. Well, we have a mature bone cell that we call an osteocyte, but we have several of the types that are very important. Osteoblasts, and to help you to make this clear, osteoblasts, blasts build. Osteoclasts break down. There are two types of bone. Compact bone, which is very, very dense, lots of osteons, and spongy bone, which is more spicules and plates, etc. And there's no real osteons there. We still do have lacunae, which are sites, pockets in there where the bone cells are residing. We have a central canal, which contains nerves, blood vessels. Caniculi are really these fine little branches, or actually fine little fissures that you see between the layers of the osteons. They provide pathways of connections between osteocytes. Oops, sorry about that. So here's an example of this. This is an osteon. Here you have the structure here with a layer of solid calcified matrix. You have an extensive blood vessel supply. You have it going right through here. This would be what we call the central canal, otherwise known as the Haversian canal. And then to move perpendicular to the long axis, we have these side connections, which are also known as Volkmann channels. And as we get deeper into the bone, we come up to an inner layer here, inner to basically right next to the medullary cavity, which contains the bone marrow. This is the spongy bone. As you can see, it's very different from, let's say, the arranged order that you have of these lamellae, the layers of calcified matrix. Okay? So this would be all compact bone. Right here is where you would have spongy bone. Now, I encourage you to take time to compare cartilage with bone. Not only the cells, the ground substance, the fibers, the vascularity, the covering, and the strength, because they're very, very important. Now, when we talk about bones and periosteum, there's a layer covering that occurs on bones. And periosteum types, you have a fibrous layer that attaches bone to the surrounding tissues and or is associated tendons and ligaments. Okay, and you can see that on the outer edge. Underneath that is a cellular layer, which functions for appositional bone growth and aids in bone repair after an injury. Okay. Now, here is the concept map. I made a simple one. I could have gotten into much more distinct differences, but at the time I was working on this. And as I said, in all these cases, these concept maps, I encourage you to print out and review. First question, are all three fiber types present in the tissue? Collagen, reticular, elastic. If it's yes, that's connective tissue proper. Okay. If it's no, does the tissue have collagen and some elastic fibers? If it's yes, then it's cartilage. Does the tissue only have collagen fibers and calcified salts present? If it's yes, it's bone. If it's no, it's blood. Now, tissue membranes are another area that we want to go over. They function as physical barriers, fascia support, and surround organs. There are different types of tissue membranes: mucous membrane, serous membrane, cutaneous membrane, synovial membrane. The one that is referred to as the dry membrane is the cutaneous membrane, because that's what we're talking about, skin. Let's talk about what the term fascia means. That's connective tissue layers that support and surround organs. Sorry about the interruption. But anyways, let me continue. Types of tissue membranes. There's mucous membranes, such things as the membrane that you see in the respiratory system, the digestive system, the esophagus, things like that. Serous membrane are membranes that you would see, for example, the membranes lining the abdomen or the thorax, right? Cutaneous, as I said, is a dry membrane, such as what you would see on the outside of the lungs. And then we have the synovial membrane. These are the membranes that exist in joints, synovial joints, things like that, knee, shoulder, hip. Fascia, you want to keep in mind that fascia is the connective tissue layers that support and surround organs. There are several different types. You want to be mindful of both the type of tissue that makes up these membranes, as well as understand the different types of the fascia. The superficial fascia you will see is comprised of both areolar and adipose. Deep fascia is dense, regular connective tissue that you would find. Subserous fascia is only areolar. And you would see these examples. Here is the examples of the tissue membranes. You have the synovial membranes, such as in the shoulder here, where you would have the epithelium, and then you would have the areolar tissue underneath this, and as well as contained by a fibrous capsule. But that tissue in between also would have adipocytes for cushioning, okay? You would also have this tissue producing some of the synovial fluid. Serous membranes, for example, you have the mesothelium, and then underneath it would be areolar tissue. Mucous membranes, that's where you're going to have the epithelium, and then the lamina propria there. And then cutaneous membrane, as I was mentioning to you earlier, you have, of course, the epithelium up here, and then you're going to have this areolar tissue, and then underneath it, the dense irregular tissue, which all of this would make up the dermis. Now, when you look at superficial fascia, that's really between skin and underlying organs, okay? When you see deep fascia, that's bound to capsules, ligaments, and other connective tissues. The subserous fascia is between the serous membranes and the deep fascia, as you can see in this diagram right here. Now, muscle tissue. The function of muscular tissue is movement of the body, transport of substances inside of the body, and the generation of heat, which is part of homeostasis, how we maintain body temperature, for example. Gets too cold, we generate our own heat by shivering. The three types of muscular tissue are skeletal, cardiac, and smooth. What you will note is that 50% of the body mass is made up of muscular tissue. This includes, of course, skeletal, cardiac, and smooth. Connective tissue makes up 45%, and then you have the last 5% split between epithelial tissue and nervous tissue, okay? Which we're going to get into nervous tissue last, but save the best for last in a minute. Skeletal tissue and cardiac have several things in common. One of them is striations. And you can see these are the condensed areas of actin and myosin filaments, et cetera, et cetera. With skeletal, though, it differs. This is more under voluntary control. Cardiac is more under involuntary control, as is the smooth muscle. What's the difference? I can basically consciously tell my body to contract my arm, etc. I do not consciously tell my heart, contract, contract, contract. That's all controlled by the autonomic nervous system, by our brainstem, etc. So in other words, this is all automated. We don't have to think about it. The same thing with when we have the contraction of smooth muscle to push food through the gastrointestinal system, or smooth muscle that is in the ureters to push urine down, propel it down into eventually the bladder, okay? Now, in skeletal muscle tissue, as I said before, you have striations. You will have the nuclei on the far edge, sort of almost on the exterior, pushed up against the cell membrane for a muscle fiber. One thing to note is that you can have multiple nuclei in a muscle fiber. That's one of the more unique ones, where you can have what's basically called a syncytium, where all the cells are basically one continuous cell with multiple nuclei, and yet that's fine for the function of the skeletal muscle tissue. You can see these line patterns here. These are the striations. With heart muscle, it's a little bit different in the sense that you get one, maybe two nuclei per cardiac muscle cell. You also have these unique connections between them, and they're what are called intercalated discs, which allow for synchronization of the contractions of all the muscle cells all at the same time, because each of the cells at the intercalated disc are connected by the connexions, and basically the structure that allows for this gap junction control that allows for the synchronization, the movement of ions across the cells to coordinate and make sure that you have one big continuous muscle contraction. You will have in cardiac muscle cells striations as well. Smooth, no striations. One nuclei per cell, and they are kind of like tapered. They're found in many hollow organs. Smooth muscles will be found in the walls of the trachea, stomach, small, large intestine, urinary bladder, ureters, uterus, iris, blood vessels, and bronchi. They are used to control either to push materials through those tubular structures or to control the flow of materials through the lumen of these tubular structures. A graphic organizer for the muscular tissues to differentiate them, two basic questions. Is the muscle tissue under voluntary control? If it's yes, then it's skeletal muscle. If it's no, then the other question is, are there intercalated discs present between the cells? If it's yes, it's cardiac muscle tissue. If it's no, it's smooth muscle. Nervous tissue. The function is really for communication with various sites of the body. Pathways to brain provide sensory information. Pathways from the brain provide motor responses. Now, it's not merely neurons. Otherwise, it would be kind of interesting to call nervous tissue just neurons, but it's not. You have also supportive cells called glia cells. The structures of a neuron consist of dendrites, axon, cell body. The functions of neuroglia, though, maintain the physical structure of the nervous tissue, repair nervous tissue framework after injury, perform phagocytosis, provide nutrients to neurons, and regulate the composition of interstitial fluids surrounding the neurons. This is key. Here is an example of a neuron. You can see that it has these dendrites which take in information. You have a large area here which we call the cell body, otherwise known as a soma, which can consist of smooth endoplasmic reticulum, a lot of rough endoplasmic reticulum, which they refer to also as Nissl bodies. Don't worry, we'll get more into this as we get into the nervous tissue in a subsequent chapter. We have a nucleus, and of course we have a nucleolus, which, as we said before, consists of ribosomes, ribosomal proteins, and RNA. We also have a lot of microfibrils and microtubules which sustain this long extension from the cell body called the axon. The contact sites with other cells, whether it's other neurons or muscle or glandular cells, etc., these are the terminal structures here. The glia cell types vary. They can be spiny glial cells, they can be astrocytes, they can be oligodendrocytes. Basically, these are involved with maintaining the function in various formats of the neurons. Here is an important area that I encourage you to print out now and retain, because it'll help you as we go through not only this chapter, but other chapters that relate to the nervous system. First off, does the cell produce action potentials? Yes, then it's a neuron. No, then the next question is, does the cell form a myelin covering around the neurons? Yes. Is the cell present in the nervous system? If it's yes, it's oligodendrocytes. If it's no, then in the peripheral nervous system, we have what we refer to as Schwann cells or satellite cells. But let's go back to the myelin covering. If it doesn't have that, does the cell wrap around the brain capillaries? If that's yes, that's astrocytes. If it's no, then the only other question you have is, does the cell act as immune cells? If it's yes, it's microglia cells. If it's no, it's ependymal cells. Now, we finally come up to just about the last section, and this is an important area. How do tissues respond to injury? When we talk about injury, we're talking about tissue damage that leads to general defense mechanisms. This can occur via exposure to toxins, pathogens. It can lead to mast cell activation and release of mast cell products such as histamine. And we face at this time the situation of infection, which is the presence of pathogens at the injury site. There's also another concept to keep in mind, inflammation, which is the swelling, redness, we would feel maybe heat, which is kind of a warmth at that site, and pain. And this is due to increased blood flow, increased vessel permeability. Now, these factors lead to several things. When you increase the blood flow, etc., you're going to have an increase in local temperature. You're also going to have an increased oxygen and nutrient supply there. That's important to begin the process not only of fighting off infectious agents, but eventually repair. You're going to have an increase of phagocytosis. A lot of times that's going to be through macrophages and a variety of other cells. They're going to be doing the fighting off of the pathogens that are infected because of this breach in the tissue. You'll also have removal of toxins and wastes. Now, these actions lead to the cleanup of the infection and the elimination of the inflammation. Then we get down to the issue about regeneration, repair. Now, fibroblasts begin to lay down a network of collagen fibers. The dense collagenous framework is known as scar tissue. Now, scar tissue is usually is remodeled and more normal tissue will appear. But what if it doesn't? That's where you have fibrosis. This is the permanent replacement of normal tissue by scar tissue. If you think about it, when you have, let's say, an exterior wound and it requires stitches, what's the purpose of stitches? Well, you've got to clean out the area of the wound, and then you begin to bring, whether it's using staples or some of the modified patches that pull the tissue together, or actually the simple needle and thread. I shouldn't say thread, it's either catgut or some type of polymer or something. The idea is to pull the tissues, edges of the damaged tissue together to allow it to build up the scarring, but then eventually to build up also replacement by normal tissue. If you do not, the tissue that replaces the normal indentation and the damage will be made up of scar tissue, which is not always as flexible as the original tissue was. Here we have an injury by a young boy, and it's a knee scrape. You have exposure to pathogens and their toxins, which stimulates mast cell activation, which releases, of course, histamine, heparin, and prostaglandins. This will begin to act on inflammation, which means an increased blood flow, increased vessel permeability. What I mean by vessel permeability is fluid in one sense or another that will be passed out. This is important to increase the body temperature there, so it increases the metabolic activity of the cells to do repairs and to fight off the infection. Increased oxygen and nutrients, which again increases the metabolic activity to fight off the infection and begin repairs. Increased phagocytosis. Macrophages and other white blood cells will be brought into the area. They will bring signals, they will send out signals to bring even more cells in to fight off the pathogens. And eventually the removal of toxins and wastes. Once we've done that over a period of days or hours, you've got a cleanup process to begin. Now, regeneration inhibits further mast cell activation. So once we've started triggering regeneration, we start having scar tissue form, which is then remodeled, and eventually you start having, let's say, below the epidermis, you have into the dermis area, a buildup of new vascularization, new tissue, and eventually you would dissolve the scar tissue slowly, methodically, and the tissue may eventually be, if everything else it proceeds normally, you will have normal, strong tissue there. If it is not done properly, the scarring tissue may be taking up the space. Hopefully, you've been able to bring the tissue together and there's been no real serious gaps so that normal tissue can be regenerated and normal tissue conditions are restored and the kid's a happy little kid running around. Everybody got that so far? Well, I hope so. Keep in mind that prostaglandins, histamine, heparin, all of these are part of the normal signals. They will act on increased vessel permeability. They will act on enhanced pain sensitivity to that area. But eventually, you have this process that begins with tissue repair. Okay? Well, that's about it. We've completed this chapter. I encourage you to peruse the chapter review section on pages 177, 178. Complete the review questions on pages 179 to 180. Complete review sheets on pages 158, 170, and 176. The next chapter we go into is Chapter 5, the integumentary system. I hope you all have a nice day.